I'm talking about a subject that is perhaps somewhat not the norm inside the association, but I think one of great interest, and I know an increasing number of us are working in this space. So I'm going to talk about traumatic brain injury, more generally called TBI, and my particular interest is in the live human brain and what happens within that. I'll show you a variety of experimental results and some simulation results and try to give you a sense of what actually happens inside the brain as far as we know. The pictures here are fairly excuse me, important. So the one on the top left, the red cells, those are cells inside your brain, which are not the neurons, which is most of what I'm going to talk about. But it turns out the majority of your brain is not actually filled with neurons, but filled with cells like these. They're called astrocytes or glial cells, and these cells are really important to the functioning of the brain. Now, within this talk, I'm not going to spend very much time on the astrocytes themselves, but they're really important mediators for how the brain operates, and we'll talk about them a little bit at the end. And of course, there's an image of the brain and the skull, as you can see it in the little graphic in the middle. The people at the bottom here, these are some of the individuals who've been involved in this work. I get a chance to present to you in DMAT, but really the people who do the work are the graduate students and the postdocs. I'm not going to, well, I guess maybe let me take the time to talk through the people whose work is presented here, just briefly, starting from the left. So on the extreme left at the bottom, that's Dr. Kshitij Upadhyay. He's currently a postdoc with me, and he's working on simulations of the brain. Next up is Dr. Shailesh Ganpule, now a professor at IIT Roorkee, who did some of the work you're going to see today. Connor Bradfield is a graduate student with me right now and is working on the mouse. I will show you some of his results. Amy Dagro was a graduate student with me, now works for the Army, for the US Army Research Laboratory, and she worked on astrocytes, which we'll talk about briefly. Dr. Yuanqiao Lu is now working at, was a postdoc and is now at Children's Hospital in DC, looking at injury problems. Jeff Gleister helped us with segmentation of so we can see where the vasculature, the blood vessels are in the brain. Professor Fatma Madhu was a graduate student with me and is a professor at the University of Kuwait. And Professor Jerry Prince continues to work with me. We are working together on MRI problems in the brain. We'll talk about that a little bit more. So Jerry works on imaging, and his images and his segmentation of images is fundamental to the work I'm going to talk about. Very good. So this is the background. So let me start with a poem. So this is by Ralph Waldo Emerson. And he says, "A man is a god in ruins." And I want to get to this idea as we go through the talk, because we're thinking primarily about injury and what happens inside the brain during injury. Let me start by recognizing something that I think all of you know, but maybe you don't normally think about this in terms of how this affects us quantitatively. So we are going to talk about humans, we're talking about people, and there's just this very wide range of people that we have. So even though I'm going to talk to you about what happens in injury in the human brain, I want you to remember that every one of us is different. We are different in our sizes, in our shapes, in our heredity, in the nutritional level we have, in the amount of sleep we have, in our current state. All of this is different. And this is a scene, by the way, from San Francisco, I believe. Just gives you a sense of the range of things we're talking about. So we look at humans, but we see a wide range of different people. We're not by any means similar. And the problems are of interest to us, the brain injury problem. You see other applications of it. So here is a woodpecker, and the woodpecker obviously spends a lot of its time hammering away at the wood. That really sends shockwaves through its head, but it seems to be doing fine, whereas we don't do so well. So if we have, most of you like the beautiful game, soccer or football, and in here we commonly do heading. Let me tell you something that perhaps is not commonly understood, but there is a fairly well-known injury that develops as a result of events like this. It's not very common, but it does happen, particularly to teenagers, where the result of hitting the ball is that you have developing blindness in one eye. So the optic nerve gets damaged as a result of this kind of impact. So most of us don't even notice it, but it does happen. And then at the other end of the scale, I want to talk about things that happen at a very small scale. So this is the blastocyst. So if you take a human egg and then you're developing an embryo, there is a point at which, and you can see it here, this is called the blastocyst, all these cells that have multiplied together, but this thing is spherical. And at some point, the spherical blastocyst is going to split, I shouldn't say split, but develop a bilateral symmetry like you and I have. So we have bilateral symmetry, right? Our left and right sides are somewhat similar. The blastocyst does not start with that, but it develops it. And that process of development is important to us when we think about the brain. All right, so I'm going to try to keep these ideas in our heads. So the skull does protect you. That's what the woodpecker tells us. You can get injury in places that you don't think about. That's what the optic nerve problem tells us. And symmetry matters. That's what the blastocyst tells us. All right, so let's talk about traumatic brain injury. There are a variety of problems where you see this. Obviously, older people falling. As we get older, we become less able to control our emotions, and we get falls. Falls can result in senility. Shaken baby syndrome. If you pick up a child and shake it, well, we do this all the time, right? We pick up our children, throw them up in the air, catch them again, they're fine. But if you shake them the wrong way, or if you shake them too hard, they can get hurt. And this is related to the problem of interest to us. Sports and impact. This is something that everyone thinks about, concussions, right? It's a big problem for us. Car crashes. So a lot of injury shows up in car. And of course, we have things like explosive blast, which I know some of us are in this kind of an association talk about. So these are the kinds of events that cause TBI, but there's a fairly wide range. I'm going to focus on mild TBI. And what I mean by mild TBI is I'm going to focus on the kind of injury that you do not easily see when you go to the radiologist. So let's say you feel you've been hurt, you go to the hospital, they take an X-ray, CAT scan, MRI, variety of different ways they can look at you, and they cannot see any injury in any of those imaging modes. That's when I'm interested. So the case of mild TBI is where I cannot see it easily in imaging. All right. So there are other problems where you see this. So this is the case of blast. This is actually an event in Iraq where a number of US soldiers were hurt as a result of blasts. And you can see the typical blast injury problem that one thinks about. So you have a major blast, you have the direct effects of the blast wave, you have the fact that the body is thrown back, then you have the fact that you hit something after it's thrown back and maybe your head is shaken. All of these can cause injury. I'm going to focus on the head and what happens with the head. But it could be as a result of blast, could be as a result of the other problems we talked about. Of course, we wear helmets to try to protect ourselves from a variety of these kinds of events. And part of the question of interest is how much can the helmet protect you? All right, so here's my big problem. I want to study brain injury in humans, but I cannot do live human injury experiments. So the classical way in which we do science, if we want to study a problem, right, we do the experiment in that area. I cannot do the live injury experiment. So how am I going to understand what happens in brain injury? Fortunately, even though I don't do the experiments, other people do. So this is an example from American football, very different from soccer. But here we go. So these people are choosing to play this game. So that person throwing the ball is called the quarterback. He throws it to the receiver, catches it, and who then gets hit. The person in the white shirt is now running back. He got hit a while ago.
Carter made the hit, and unbelievably, the Kings secondary gets torched there.
And now he falls.
And remember, Bush has all.
So he got hit at one point, was fine, continued to run for a few seconds, and then realizes that he's injured and falls. So my interest is in what happens during that event. Now, as you saw in this case, we've got a variety of cameras watching such an event, so I can actually reconstruct the event. And using that reconstruction, we can figure out how injury occurs. But remember, this is DYMAT, right? So our fundamental interest is dynamics. So let's think about dynamics. We're talking about dynamics of a structure, which is the human head. So we need to understand that structure. Let's talk about the basic anatomy. So if you look at the head, as I said, we are bilaterally symmetric. If you look along the midplane, so imagine a cut right down the middle, right? And look at one side compared to the other, this is what you'll see inside the head. You see the skull, you see a membrane or set of membranes called the CSF and the dura, and then you see the brain. Most of that is what we call the cerebrum. Part of the brain is the cerebellum down there, and then you have the brainstem, which goes down and then goes through, becomes the spinal cord going down to the rest of your body. You have these membranes that separate different parts of the brain. There's the tentorium, which separates the cerebrum and the cerebellum. And then there's another membrane that separates the left and right-hand sides of your brain, and that's called the falx. So these things matter, right? So you've got a structure in there. We'll see that in a little more detail. So here's a picture of the brain looking from the top down. There's a falx in the middle, and there's actually a stiff membrane inside there that separates the two halves. Not all the way, but most of the way down. So this is the structure we're looking at. The boundary conditions are applied to the whole head. The injury occurs inside the brain. We are trying to understand what happens inside the brain. Okay. So we are understanding what happens inside brain tissue. So what's the structure of brain tissue? This is a sketch of length scales going from 1 meter on the left to 1 micron on the right. Your head sits at about 10 centimeters or so. So there is a section of the brain. You can see the left and right hemispheres, if you like. You can see the connections between the left and right hemispheres. That connection is important. If you look a little closer at the tissue inside the brain, you'll find a variety of neurons. These neurons have axons, which are these cables moving down. And if you look at a given neuron, this is what it looks like. It's got a cell body, an axon, and connects to other neurons. So at the micron scale, we've got these individual cells. Collections of these cells form tissues, and a structural collection of this tissue is the brain. And then the brain is housed inside the head. So that's what we're interested in. So the injury, let's think about this. The impact occurs at the head level. So this is at the 10 centimeter level. But it turns out the injury occurs at the 1 micron level, at the cell level. So we're going all the way down from the head to the cell. So one way to think about this, the boundary conditions are applied on the left, injury occurs on the right, but behavior occurs back at the left. So because of the injury, you get changes in behavior which move back to the left, to the larger scales. So how are we going to do this? Trying to understand how to do dynamic brain biomechanics. So we will take an event where somebody gets hurt, and then we'll reconstruct the event. So we've got all the cameras, so we know what the motion was. We can track all the accelerations. We can even put accelerations and accelerometers in the helmet. We then build a model for that head, for that individual's head. We simulate what happens inside the head during that event, and then we predict where injury will occur. So we will identify not just what happens, but where the injury is likely to happen inside the brain. This is our interest. Okay, so you can see pictures on the right that show different parts of the brain that we can identify and which part connects to what kind of cognitive function. All right, so that's big picture. So we have a problem where we have humans getting hurt. Sometimes they choose to play in these kinds of events. We can track that information, we can simulate the condition, and we can go through. The big problem is this. The first question is, I need to know when will you get it. So that's the safety threshold, right? If this happens, you're likely to get hurt. If this does not happen, you're not likely to get hurt. So what's the threshold? The second question is, where will you get injured? So it's not just when. So assuming you're going to be injured, where will you be injured? Which part of the brain? That tells you what the likely consequence is going to be, what kind of behavioral change there'll be, and what kind of treatment you might have. And the third question is why does the injury occur? Now, this is much more of a cellular problem, and that tells you what drugs to use. So today I'm only going to talk about the when and the where. I don't have time to talk about the why, but these are the three questions you have to answer. So when will you get injured? Where will you get injured? And then why? I'm not going to talk about the pharmaceutical side at all. So let's start with the when will you get injured. We need some kind of an injury threshold. We want to be able to look at an event and say, this person's going to be hurt. We've got a variety of these thresholds already identified right now, and you can see some of the injury thresholds on the right, these equations on the right. The head injury criterion, gambit, hip, brick. Each of these relates to how much acceleration you've got, how long the acceleration's been on, what kind of acceleration, is it linear, is it rotational, and then the motions of the head. That's typically how these are done. My point is all of these injury measures are based on the whole head. They look at the motion of the entire head and they estimate when injury is likely to occur. None of these are particularly good in the sense that you can have all kinds of motions that can cause injury that don't necessarily show up in one of these criteria. But all of these are fairly good in the sense of giving you a critical condition. Okay, so they're good for critical conditions, but not good for the detail. All right. So the way we are going to think about this, because we're mechanics people, is we build a multi-scale view of the brain. So we say, here's the brain. We've got the organ on the left, that's the whole brain. That's got tissues inside it. The tissues have axonal bundles, so bundles of these fibers that run together. Those are about 100 microns across. And then we have individual axons. So here's an image of parts of the brain. You can look at parts of the tissue. You can look at the fibers bundled together, and then you can look at individual axons. So you can build all of these up. But we want to do the mechanics. So we're going to be sitting on that top level. We've got to build a way of thinking about this. So we will do imaging, magnetic resonance imaging, MRI, to understand the structure of the brain. We'll do diffusion tensor imaging, another kind of imaging, to understand the tissues in the brain and the orientations in those tissues. We'll study the material behavior of samples of these tissues by taking it to the lab and doing the experiments. And then we'll build an axonal strain injury criterion by measuring when an individual axon is injured. So let's start with the first one. As always in DYMAT, the first thing we think about is materials. So what's the material model? And we are going to think primarily about the white matter in your head. That's basically where you get injured. So the first thing to recognize is brains are soft. So our brains are generally soft, squishy, and fibrous. And let me run this movie so you can see it.
This is the right hemisphere of a human brain, one of hundreds of brains donated for medical research in the UK every year to one of 10 brain banks. Now, if I pick it up, intact, the human brain weighs around 3 pounds or 1.5 kilos. It makes up just 2% of the body's weight and yet consumes 20% of the body's energy. It contains 100 billion nerve cells.
Okay, I'm going to stop that movie just so I can talk about the one of the things that the movie doesn't show. So in the movie, when you see that person holding the brain, that brain looks pretty stiff. The real brain is nothing like that. It's much softer. If you try to hold it in your hand, it will tend to fall around your hands and moves really quickly. It's very squishy. What that means is that the stiffness is low. If the stiffness is low, that means the wave speeds are low. Thinking about the stress waves inside the brain. The pressure wave is fast because it's nearly incompressible. So the pressure wave speed is about 1 kilometer per second. The shear wave is slow because the shear stiffness is so low. The shear wave speed is only 10 meters per second, actually less, a few meters per second. So the end result is that when you have an impact, the pressure wave moves really quickly through your head. So your head equilibrates in terms of pressure really fast. But the shear wave moves much more slowly. The time it takes for the shear wave to establish itself in your head is around 30 milliseconds. Most of the injury is due to the slower shear wave. Rotations dominate the injury more. Okay. So we can go in and measure the properties of brain tissue. So here are two kinds of experiments you might do. These are just using Kolsky bars, what some of you call split Hopkins and pressure bars. In the picture on the top, what we're doing is measuring the volume change of the brain as a function of the applied pressure. It's basically a uniaxial strain adaptation of the Kotsky ball. And that gives you the kind of plot you see on the right. You can measure the pressure volume behavior of the brain, and you find that the bulk modulus is about 1.6 to 1.8 GPa. So it's not very stiff, but it's comparatively stiff, and I'll tell you why in a moment. On the bottom left, you have the shear wave behavior, the shearing behavior. So you build a double lap specimen and then you shear it dynamically. You can measure the behavior. You'll find that the shear moduli are kilopascals, maybe 10 kilopascals. So 10 kilopascals versus 1 gigapascal. So that's a huge difference in orders of magnitude in stiffness. And that's why these wave speeds are so different. All right. So we know the properties. We can now build a constitutive model for white matter after having made those measurements. So what we do typically is a nonlinear elastic or nonlinear viscoelastic behavior. So the equation on the top, that's the strain energy function you might use. So I1, so the way to think about this equation is it's telling you what the elasticity is of the brain. The term on the right is volumetric, that's the hydrostatic term, and the term on the left is shearing. The shearing term has different components to it related to the orientations of the fibers in the brain, because the brain is not isotropic. So you can handle the anisotropy, and you can look inside an MRI or a diffusion tensor image and actually calculate the anisotropy, and that's what this number kappa on the bottom is. It tells you how anisotropic the brain tissue is, the given piece of brain that you've got. So those are the experiments we're doing on small pieces of brain, a few millimeters in diameter, but the whole head is now 10 centimeters across. All right. So we have a constitutive model for different parts of the brain, but we want to use it for the whole head. So the way we're going to evaluate the model, decide if the model is any good, is we're going to use live human brains using volunteers. So this is the result of a series of grants that we've got from the National Institute of Neurological Disorders and Stroke that looks at injury in the brain and the movement of the brain. These are the people who are the PIs of the grant. Phil Bailey is actually the lead at Washington University in St. Louis. Zoom Farm at Henry Jackson and NIH does a lot of the imaging. Jerry Prince does a lot of the MR tagging and the segmentation. And so, of course, we have our students and postdocs who work with us to do this work. So we are doing 3D live human brain measurements. And here's how it works. So you take a volunteer, you put them inside an MRI, a magnetic resonance imaging system, and you ask them to turn their heads. So they're turning their heads themselves. We are not turning their heads for them, they are turning their heads. When they turn their heads, eventually it stops somewhere. It can only move so far before it stops. And during that time, we're taking images with the MRI, and we are able to measure the motion of the head. So let me run this again. So we have tagged images inside the head. Basically, what you're doing is you're using the MRI to generate signal inside the head where different parts of the brain can be identified differently. It's a method called tagging, related to what you're doing with the protons or with the hydrogen atoms. But the end result is that we can measure strains in the brain in 3 dimensions during the event in the live human brain. So this is a really significant capability. 3D dynamic brain measurements. At relatively low speeds, the MRI is operating at 8 milliseconds or so. So this is how we do our work. On the top left, you have an anatomical scan. So first, you as a volunteer will come in and we'll scan your brain. And we'll get images like what you have on the top, the T1 image, the diffusion tensor image. And then the bottom left, we do a biomechanical scan. That is the same person after we've scanned your brain. We come in and say, okay, now we want you to move your brain, move your head inside the MRI. You move it yourself, and we will measure how it deforms. And then with that, we can build a subject-specific 3D head model. What I mean is, I build a separate model for every individual's head, for every subject's head. So it's subject-specific. So there's a volunteer moving his head, and I build that model, and now I can run a simulation using the material models, material properties that I have measured in the lab, and I can calculate what's happening inside the brain, and then I will compare that with what the measurements are. So I measure three-dimensional strains in the brain in 3D. I can calculate them and compare the two. So here's how it works. This is how you do the tagging. So we actually do two different kinds of motion. We do head rotation, where you're doing this, so moving left to right, and then we do head extension, where you're doing this. It's like nodding, right? So you're moving your head up and down. And in each case, we can tag the brain and measure how much motion occurs. So if I'm doing both together, my temporal resolution is 18 milliseconds. My pixel resolution is 1.5 millimeters inside this. All right, so let's see how this works. I have my MRI imaging, which is the top left. I can segment that, that is, I know what the different parts of the brain look like in the MRI, and then I end up with a segmented version on the top right. So basically, I'm able to name each part of the brain. I can do diffusion tensor imaging, which tells me how water flows in the brain, which tells me how the fibers are oriented in the brain. So how diffusion occurs in the brain, not really water flow. That gives me the orientation distribution, and now I build a model for that individual's head. So I've got a whole head model now for this individual. All right, I'll run a calculation now using the material point method and a software system called UInter, a software framework, which lets us do very large-scale calculations. So for each human, I can do a full-scale calculation of their heads. It takes a good bit of time, but I'm doing here 260,000 material points inside this one head. The nice thing about this method is I can take voxelated information from the MRI and directly import it into the simulation. Here are the material properties that I use. You can find it in that paper. This is recorded, so you'll be able to get the material properties yourself. These are the properties we're using for live human brain. All right, now here's the boundary condition and the loading. So I measure the acceleration as a function of time during the MRI experiment. That's the picture on the right. I then run the simulation on the left by applying that boundary condition, and I can see the motion in the simulation. I could also see it in the experiment. So now I can compare the two. Let me start by showing you what the simulation shows. So the way to look at this picture is on the right-hand side, the columns are individual times. The rows are sections of the head, so different levels of sections of the head. So as I run the movie, so this is a calculation. What I'm showing you is the shear strain. You can see the shear strain propagating into the middle of the head and then propagating out. It changes sign as it propagates in and out. It takes about 30 milliseconds for the shear wave to propagate in and then propagate out. So there's a natural frequency for the brain then. Okay, but given that 30 millisecond time scale. And then you can see the images that we have for each section from the simulations on the right. So this is all simulations. But we can compare that to experiment. So here are some experiments on the left and simulations on the right. So this is looking at the radial transverse strain and one particular strain component. We're looking at different amplitudes for different times. And the comparison is not too bad. It's not great, but it's not bad. This is actually one of our big problems, is deciding when is a simulation good enough, because you're doing three-dimensional data, which is three-dimensional displacements and strains as a function of time. If you match at this time, it doesn't mean it matches at a later time. How do you compare these things? Well, that's a problem I don't have time to talk about, but it's really important for it. Of course, you can compare individual things, compare the experiment, the simulation for displacements in different directions, compare the strains in different directions, and it's not too bad an approximation. So I would think of this as validating the model. I'm not using this to predict anything yet. I'm just comparing what my material properties said, and when I put it into the simulation with what I saw in the live human individual, right? It's a pretty good comparison. Okay. So this looks like a reasonable model. Now I should be able to go look at injury. Before I do that, let me point out something. Each of us is different. So here are 6 different people. For each of them, I'm showing you the experimental results on, sorry, these are the simulation results I'm plotting here. And you see that every one of them behaves differently in terms of the same motions. Inside your head, the different parts of your brain, these are all different sizes, different shapes. And so you have different levels of sensitivity to injury. So subject number 6 for the same motion is seeing much more deformation than subject number 1. And so subject number 6 perhaps should be a little more careful about certain things. This subject-specific behavior is really important to us. All right, so now we have a model. We know how we can work on it for individual humans. Now we're interested in injury. How do we get to injury? So this is just an image of a movie that shows you the different parts of the brain. So here are the ventricles. These are the parts of the brain, essentially gaps in the brain through which fluid flows. On top of that, as this thing runs, you will see this is the cerebellum at the bottom. As you keep going, we'll be able to add the cerebellum. This is mostly white matter. And then we can add the vasculature, which is the blood vessels of the brain. There's the brainstem. And there's the vasculature, the blood vessels. So all of this is in your model. And then you're trying to calculate when injury will occur. You can see it's a very complicated structure. But that's the structure we have. That's what our brains are like. All right. So you can run calculations on individual events. Like I started out, I said, there's an event we can observe on the sports field, and then we can calculate what's likely to happen for that individual, run the calculation. Now, one difference is, up to now what I showed you was I took a given individual and I built a model of their head. For the sports event, typically I don't have a model for that individual's head. So I have to take a generic head model and run a calculation using that. But I can predict when axonal damage will occur based on the idea that there's a critical axonal strain which will cause injury. And this will tell you which parts of the brain are likely to be damaged as a consequence of that. Because each part of the brain has a certain level of importance with respect to certain kinds of behavior, you can then predict different kinds of behavior and ask, does that compare with what people observe in the field? And it works reasonably well, but not great. So we still need a lot more data on that. So the basic point I'm trying to make here is, in humans, yes, I can do the calculation for injury, but I'm really limited by the fact that I don't have the data on that particular human in terms of that particular person's head and his brain, or her brain. So instead, what I'm going to do is do some experiments on mice. So I'm going to use the mouse as an animal model for TBI. Now, the thing about the mouse is it's really small, so the brain is really hard to work with. On the other hand, I can get lots and lots of mice. The other thing about the mouse is it's very well studied. We have a whole range of information on the mouse. The mouse brain is distinctly different from the human brain in some ways, though. It doesn't have those folds that you're used to seeing, for example, so it's a lissencephalic brain, not a gyrencephalic brain. So let's think about how we connect from mice to man. So here's the human case, right? Basically, our argument for the human case is if I know when a piece of the tissue, the axon, is going to get injured, right? A given cell is going to get injured, I can say something about injury in the human. The mouse is similar, right? The mouse has a certain size, it's got a certain size brain, it's got tissue which is very similar. So my argument is basically, I'm going to study injury at the tissue scale for these two and do the experiment at the whole mouse scale, and then try to calculate what's going to happen at the whole human scale. So here's a mouse model. You take the whole mouse, you can build a model for the mouse. This is due to the Army Research Lab and Amy Degro. So you then have a model for the mouse brain. In the mouse brain, you can track the fibers. My colleague Vasily Kolyotsis at Hopkins in pathology gives us the fiber orientations using Clarity. So we have all of that information. And then we do these kinds of experiments. So on the left, we do drop tower experiments, which will allow us to see the motion in the brain. On the right, we do simulations, and then we bring those together to understand mechanisms. Okay. So here's how we do in situ measurements of mouse brain deformation. So we take a mouse head, we cut it in half. We then take half the head and put it inside a structure, basically a little carrier. We take that carrier, cover it with epoxy, but we can still see it, so we can do DIC on it. We put the whole mouse, this half mouse head, inside this system where we can now rotate the mouse head really fast, and then I can measure using DIC how the mouse brain deforms. I do this for different rotational speeds, 100 radians, 150, 200 radians per second, and so forth, and then I can measure the deformation in each case. I also have a model for the mouse head, so I can now compare the two. So what do you see? This is a half mouse head when it's rotating. You can see the strains and you can see the displacements. I think I should be able to get this one, the light to move. Maybe that's not very good. There are the displacements. You can calculate the strains as well, right? So once you have that, you can compare that with the simulation. So here's the experiment on the left, there's the simulation on the right. And we begin to understand some things about how the structures in the mouse affect the deformations of the brain. All right. So let me just summarize a few critical issues here. The first question is, what is the injury threshold? When will you get injured? And my argument to you is that the classical threshold measures, which relate to forces, impulse, acceleration, these are head scale measures, which are reasonable for broad application, but really don't tell us enough about individuals and what's likely to happen to you as compared to the generic person. Most of the data you have is on people who are 18 to 25, 18 to 30 years old, and most of the people who are injured are not that age. So we have to figure out how this works. We have to relate macro scale measures of anatomy to the neuroanatomy. That's what we are trying to do. The symmetries matter. Whether you move this way or this way, how you rotate your head makes a big difference. The animal model is actually very useful because it tells us what the important pieces are, and we can then work our way back for the human brain. All right. I'd like to stop there. I'd like to acknowledge a few people. So this is all funded by, or mostly funded by the National Institute of Neurological Disorders and Stroke. There was some funding for a couple of the students from the Army Research Lab as well. It's funded through R01, R56, and Euro1 grants, particular kinds of grants. Several people that have really helped us, people like Adam Fournier, Shailesh. I talked about some of the others, the Uinter software suite, with support from Jim Gilkey and Don Parman. That's been really critical for us to do the calculations. On the mouse experiments, Jeremy Rosen, Vasily Kolyatsos and Nicholas Ziodas for pathology, and Susumu Morin for radiology. So that's the basic story, and I want to leave you with a movie. So this is the mouse. This is the kind of experiment that is typically done on a live mouse. This is when the mouse is injured, right there at the top, not before that. Incidentally, from this particular experiment, the mouse got up and walked away. All right, let me stop there, and